We made it. We made it to the end of the textbook, and we are now at chapter 17, the final chapter. And this chapter focuses on, first of all, cancer, what it means to have a cancer, and how the immune system looks for cancer cells and fights off cancers. And then we'll spend a little time digging into how we can harness the power of the immune system to develop cancer treatments. So just a little bit of background to get going. At the time of publishing of this text, the data of different types of cancers is shown here. So cancer is a big collection of diseases that have to do with abnormal and invasive cell proliferation. Whichever cell line is proliferating and moving into other tissues, that determines what type of cancer an individual has. And for a cell to become a cancerous cell, a few things have to occur. There has to be mutations that accumulate within the DNA. Those mutations have to be involved in cell cycle, cell replication of some sort. There needs to be a failure of repair mechanisms. So the cell has DNA repair mechanisms. So if mutations do occur, they can hopefully be caught and repaired before that cell goes on through division. Sometimes those mutations occur in those repair enzymes. And then a third line of defense is apoptosis of an abnormal cell. And somehow the cell that has now had these mutations escaped DNA fixing those mutations, somehow gets away from apoptosis or programmed cell death for being out of control. And again, at the time of publishing of this text, there's about 18 million new cases each year in the US. I believe this is US data. I'd have to look at the actual footnote in the book. And about 9 million, or a little over 9.5 million deaths a year. And we see a major type is lung cancer, followed by breast cancer. These are the types. Prostate cancer, rectal cancer, colon and rectal cancer, and then a variety of others, where lung cancer is more lethal, and then followed by either colon or rectal cancer and stomach and liver and so on. So lots and lots and lots of people are affected by cancers, either have the disease themselves or you have a loved one or know somebody that's had it. To the immune system, though, cancerous cells look different, much in the same way that a virally infected cell looks different. And essentially, the cell that is cancerous has abnormal function. It's not normal. It's not normally producing what it needs to produce. It's not normally functioning the way it's supposed to. And it's definitely not dividing at the pace that it's supposed to. And because it looks different, the immune system can then attack it and remove it from population without, hopefully, before it can develop into a tumor and spread to other tissues. And so the immune system is very, very important and central in preventing cancers from developing in individuals. So most cancerous cells that an individual ends up developing are eliminated by the immune system, and you never know that you ever had a cancerous cell to begin with. And it's only a few or a minority of those cancerous cells that are actually allowed to progress to full-on disease. Once somebody has cancer, there's a few different ways it can be treated. The tumor cells or cancerous cells can be surgically removed. They can be killed off with radiation. Radiation is going to attack any rapidly dividing cells. And then chemotherapy or drugs, cytotoxic drugs, can be given to try to eliminate rapidly dividing cells. These treatments can cure a lot of patients, but not all. And if it might be able to make somebody cancer-free for a certain amount of time, but it may not be able to eliminate all the cancerous cells in the body. And cancer immunologists are very it's a very hot area of research, trying to find ways to get the personal immune system, to kill their own personal cancerous cells. So harnessing that power to wipe out cancer in a very, pretty safe mechanism, at least safer than what we see with chemotherapy or with radiation, or even surgery for that matter. So the way this chapter breaks it down, I kind of already talked about it, but 3 major pieces, right? We're going to start out in this lecture. We'll talk about the process of cancer development. So a little bit of background cell biology to just look at what makes a cell become cancerous, and then what a cancerous cell looks like. And then in the second lecture, we'll move into the last 2 components of this chapter, where we talk about how the immune system normally responds to a cancerous cell, and then look at how the power of the immune system is utilized or harnessed to develop immunotherapies to help treat cancer. So that's how it's going to be divided. So we'll just start out with a little bit of background on cancer. Now, if you've taken a cancer biology course or a cell biology course, a lot of this is probably going to be review. But at the most minimalistic, basic level of describing what a cancer is, cancer comes about because cell cycle control is lost. A cell starts dividing out of control, and it's called uncontrolled growth. So the body has to grow, whether it's to grow in size, like being a baby up to an adult body, or replenishing or replacing dead cells. There's always got to be some sort of cell renewal occurring. Some cells renew faster than others, and some don't really renew at all, or very little, or very slow. And so DNA replication is an essential part of life. When a cell is getting ready to divide, it will go ahead and start to go through mitosis, and it will make sure that all of the DNA is opened up, read. It's going to create a whole new set of DNA, divide the nucleus into 2 with matching DNA, and then cytokinesis occurs, and that's normal. And there's a lot of enzymes that are involved. And so if you've taken a cell biology course or a molecular biology course, you know that there's a whole slew of enzymes that are going to be opening the DNA, copying the DNA, proofreading the DNA, putting it all back together, packaging it. And essentially, there could be a mutation in any one of those genes. And every now and then there can be an error that slips through. And when we see an error occur in DNA replication and it changes, maybe a nucleotide gets substituted for a different nucleotide, maybe there's a switch or inversion, or maybe a chunk is missed, whatever it might be, that's called a mutation. Any type of change in the DNA is called a mutation. Now, if it occurs in a sperm cell or an egg cell, we call that a germline mutation, because that has the potential of being passed down to progeny. If it's in any other type of cell, we call it a somatic mutation, because it's only going to affect the individual in which that cell is found. It might never even be noticed. So if mutations then affect the genes that are responsible for cell cycle control, say either the DNA replication or the DNA proofreading, that can lead to a cell losing its control of its cycle and divide and divide and divide and divide again. And then you end up with an uncontrolled proliferation, which can lead to a whole bunch of mutant cells. And eventually those mutant cells can move from that initial tumor into another area of the body and start affecting the physiology. And then we see this as the disease called cancer. So a few terms that go along with cancer, I've used the word tumor. Tumor actually is just swelling. And so you can have a cancerous tumor, or you can have a benign tumor, or a malignant tumor would be another way of saying cancerous, in that it's continuously increasing in size and can metastasize or move to a different location of the body. Or a tumor, still swelling, It can be benign. And so those cells are not really malignant or cancerous, and that tumor is going to grow, but it's never going to move to another location. It's going to stay in the area in which that cell's dividing, and it's probably going to be limited in size. It's not going to grow exponentially forever. Now, a tumor and a neoplasm is a new growth. And so together, both tumor and neoplasm are going to describe situations in which cells or tissues are going to be increasing abnormally at an abnormal rate. The branch of science that studies cancers is called oncology, and onca is cancer. That's where, so the study of cancer is oncology. And then, cancer is the disease caused by these tumors that are malignant and are able to move to different parts of the body. A little bit more for terminology: metastasis or metastasizing. That's movement of cancerous cells from the original tumor out into other tissues. And then those cells can, once they're in their new tissue, can start to initiate growth of an additional tumor. So this can happen especially. So cancers that a lot of opportunity for DNA to replicate, where there's a lot of cell turnover. And so every couple days or maybe every few hours, the urogenital tract, the mammary glands, those are all areas there's going to be DNA replication, which allows for a chance that something could go wrong. Now, the next set of words here are based on what type of tissue the cancers arise in. So if it's a carcinoma, that's going to be an epithelial cell. If we see a sarcoma, that's going to be every other type of cell outside of carcinomas. Leukemia, except for leukemias, are specific to just blood group cells. So circulated immune cells, lymphocytes, maybe a few other types, but mainly lymphocytes. Then we have lymphomas. That's actually going to be a cancer of the lymphatic tissue itself rather than just the lymphocytes. And then a myeloma is going to be a cancer within the bone marrow. So these are all different types, and then you can throw other words in front of these to better describe what type of cancer an individual has. So here are the tissues that give rise to cancer most often in the US population. And if you take a look at those locations, it's definitely places where you see a lot of cell turnover. In males, you can see that prostate cancer is a common place for cancers development. In females, you have breast cancer, but also ovary and uterine cancer as well. Otherwise, then we see across males and females bone cancer and lung cancers and skin cancers and all of those. But you're going to have a higher rate of turnover of cells in these types of tissues. Okay, it's important to know that it's not just one single mutation, though, that typically leads to a cancer. It's actually going to be multiple mutations that accumulate over time. The cell evolved in a way that to lead to a cancerous cell. There's a lot of checks and balances in place and a lot of different enzymes. And so there's never one protein that's going to control all of cell function. And that's good, because then that means multiple mutations have to accumulate across multiple different genes in order for a cell to actually become cancerous. And the chance that one cell would accumulate all these different mutations is much smaller than if it was all focused on one single protein. And so a malignant transformation is a term that we use for when a cell begins to, or moves from the point of being a normal cell into a cell that can form a cancer. Now, these genes kind of can be grouped in the genes that are groupings. We can call them proto-oncogenes, or tumor suppressor genes. And these are the main groups. If mutations accumulate in these two groups of genes, that's when cancers can come about. Does, because it's like, yeah, you would want to have genes that are going to suppress tumors, right? And so P53 is a major example of one of these proteins. These are genes that are considered tumor suppressor genes, are going to code for proteins that are in charge of preventing unwanted proliferation. So P53 comes in there to make sure that a cell's not going to divide outside of or out of control. And if it does find a cell that is losing cell cycle control, then it can program that cell to die by apoptosis. P53 is very important and most notable because we see that P53 mutations in about half of all cancers that are out there. So it's a much more common mutation associated with cancers than most other genes that we see. So that makes sense. Now, the proto-oncogene is like, why would you want a set of genes that are pro-cancer, right? Because proto-onco would be like pro-cancer or pre-cancer. But it's not that they're pro-cancer, it's just that they're the genes that if they get mutated, then they can lead to an oncogene. And so then we can call it an oncogene that leads to cancers. So these proto-oncogenes code for proteins that are part of the normal cell cycle. And there's a lot of them, more than 100 of these different genes, proteins that are involved in cell cycle control. And they're going to be things like controlling the cell growth, growth factors, signal transduction, anything that's going to be important in telling the cell or giving the cell signals to go through a full DNA replication, mitotic event, and then cytokinesis. So then when one of these or multiple of these proto-oncogenes become mutated and end up developing a cancer, we call it an oncogene. So those are the two groups. And typical progression, what happens is there's a normal epithelial cell. It has normal genes, no mutations that matter. And all of a sudden there is a mutation in a tumor suppressor gene and the function of that protein is lost. Okay, not so bad. The epithelial cell might proliferate a little bit more than normal, and then that means that there's more chances for DNA to have errors. Well, then an oncogene gets mutated, which then allows for the small growth to start to become a tumor. But it's really not cancerous yet. It's just growing at that one location, so it's a benign tumor. But it's replicating over and over. And so another mutation happens in another tumor suppressor gene, which then will allow that protein to be dysfunctional and lead to that benign tumor now being able to become larger and larger and larger. Well, then all of that replication can lead to errors in DNA replication. And P53 protein becomes dysfunctional due to mutation, and then the cell cycle is completely lost. The tumor can replicate out of control and start to spread throughout the body. And so we have a whole bunch of mutations that can start to accumulate, which would then lead to metastasizing or metastasis of this tumor to other locations and the disease called cancer. So it isn't a one single event, but rather accumulation of several smaller events that can lead to the development of cancer. Now, these mutations can speed up, not just are they due to DNA replication, but if people expose themselves to what we call mutagens or carcinogens, that can lead to extra DNA damage and extra amounts of potential mutations. So it can be genetic factors, it can be environmental factors that can influence the increased risk of mutations to be accumulating. So for like familial or hereditary type deals, this would be if a mutation is in those germline cells. So a sperm or an egg has some sort of mutated proto-oncogene or tumor suppressor gene that gets passed down to the progeny, and then this new child has a predisposition of having a cancer because those genes are already mutated. But environmental factors like exposure to certain chemicals or environmental pollutants can lead to increased mutations as well. When something is able to physically or chemically alter the DNA, we call that a mutagen because it is actually mutating or changing the DNA. So chemicals, UV radiation creates thymine dimers in DNA. Other higher levels of ionizing radiation can lead to breaks and changes in the DNA, and that would be a mutagen. A carcinogen is going to be one of those mutagens then that causes mutations that lead to cancer. Cigarettes are probably the most notable for having carcinogens that people inhale and can lead to lung cancer. And then sometimes viruses or an infection with an oncogenic virus can actually cause a cell to lose control of its cell cycle and transform it into a malignant cell. HPV is probably one of the most notable viruses that's an oncovirus because it can alter the cervical cells to a point where they can become cancerous. So that's where I want to stop then with this first lecture. Again, it kind of gives us a basis as to how cancers develop, what cancers are, and then what a cancer looks like to an immune system. And that's what we'll pick up in the second lecture.